got the 20.
Welcome to ECYT's conference call to discuss the company's operations update. Participating on the call today will be Mike Sherman, President and CEO, Dr. Allison Armour, Chief Medical Officer, and Michael Andriole, Chief Financial Officer. At this time, all participants are in a listen-only mode. Following their comments, we will open up the lines for questions. Please be advised that today's call is being recorded and webcast. During this conference call, the company may make predictive statements concerning future events or developments. Actual results may differ materially from those indicated by forward-looking statements. Please refer to ECYT's filings with the Securities and Exchange Commission for a discussion of risk and uncertainties impacting those results. Now let me turn the call over to Mike Sherman.
Thanks, everyone, for being on the call with us this morning. We are really pleased to announce that we've obtained an exclusive worldwide license from Advanced Biomedical Compounds, or ABX, in Germany for PSMA 617, allowing us to develop lutetium PSMA 617, a late-stage PSMA-targeted radiopharmaceutical therapy. In addition to our press release this morning, please refer to our Investor Relations tab of our website, where we've included a presentation which summarizes this therapy and also the impact of this transaction on the company. Most importantly, we view this transaction as transformational for patients. It accelerates a novel and needed new therapy to prostate cancer patients. It's also transformational for ECYT. Accelerates our path to potential registration with a product that fits seamlessly with our strategy and our capability. I'd argue the word transformational is overused by management teams. In this case, the word is absolutely fitting. Based on the activity of this drug has demonstrated in hundreds of patients already, we believe it has the potential to be a life-saving treatment for a significant population with a critical unmet medical need. Recent results reported at the ESMO meeting in Madrid in heavily pretreated metastatic castrate-resistant prostate cancer patients reflected 57% of patients experienced PSA response and a RECIST response rate in soft tissue disease of over 70%. This is a drug that has been deemed so important by physicians and prostate cancer patients. It's been used to treat well over 300 patients, primarily through investigator-initiated trials in the compassionate use setting without formal sponsorship from a drug development company. Our hats are off to those at the German Cancer Research Center, DKFZ, and the University of Heidelberg and ABX for their vision, discovery, and development work they've led to date. We at ECYT couldn't be more pleased to make the investment to accelerate the development of this drug for prostate cancer patients around the world. This transaction leapfrogs ECYT into a leading position within the emerging field of targeted radioligand therapies. It accelerates our near-term catalysts and positions us with the most advanced PSMA-targeted radiopharmaceutical in development for prostate cancer, representing a revenue opportunity of more than $1 billion. It's rare that you can look outside of your own development pipeline and find an asset that is this compelling in terms of activity that fits so well within the capability and experience of the company. In this case, our experience in targeted radioisotopes with our own imaging agents, our experience targeting PSMA, including the process of selecting PSMA-positive patients for therapy, our knowledge of the prostate cancer space, and strong relationships with the leading physician investigators around the world positions us as the preferred partner to advance this therapy. While our focus will be on the development of PSMA 617 for now, there is longer-term opportunity here to build a pipeline of radioligand therapies targeting other diseases. While there have been advancements in therapies for prostate cancer in the last several years, once the disease becomes resistant to hormone therapies and spreads beyond the prostate into bone and other organs, it's essentially a death sentence for many men. Close to 300,000 men worldwide die annually of prostate cancer, with the number expected to grow to over 400,000 in the next decade. Currently available treatments provide only marginal benefits to a small number of men in this advanced stage, and these treatments do not provide a meaningful extension of life. A change in the treatment paradigm has been occurring recently as oncologists have begun prescribing late-stage treatments in earlier stages of the disease. This change is resulting in a gap for later-stage patients who have progressed on these therapies and their disease is now resistant to them. Lutetium PSMA-617 has the potential to address that gap. While PSMA-617 is a first-in-class radioligand therapy, there has been a relevant path paved in prostate cancer through the commercialization of the radiotherapy radium-223. This includes the development of the referral networks and, in some cases, the integration of nuclear medicine capabilities within urology practices. Despite only addressing disease that has spread to the bone, patients with soft tissue metastases are contraindicated. Radium-223 annual revenue is expected to exceed $400 million this year and continues to grow rapidly. PSMA-617 has the potential to address a much broader patient population, those with bone-only disease, in addition to those with bone plus soft tissue disease who are positive for PSMA. Most patients who've received PSMA-617 have been treated through investigator-initiated trials. Under these trials, the protocols of the drug has been administered with multiple dosing schedules and treatment regimens across patients with varied history of prior treatments. And the results have been strikingly consistent across each of these studies. Even more compelling is the recent prospective trial Dr. Michael Hoffman recently reported at the ESMO conference in an oral presentation in front of a large crowd of the top prostate cancer researchers in the world. His data demonstrated even more compelling activity, likely the result of his refinements to the patient selection criteria. Allison will highlight that data more in a moment. For the past couple of years, we've been focused on addressing the unmet needs of prostate cancer patients, and lutetium PSMA-617 will allow us to help many of these patients more quickly and in a more profound way. Based on our deep relationships with the physician community, we've seen firsthand their incredible excitement around this therapy. We've already begun dialogue with these investigators on registration strategies for this agent, and we're excited to move those discussions forward with regulatory agencies. Leveraging our significant technical expertise with PSMA targeting and critical regulatory expertise in the co-development of diagnostics with therapeutics, we believe we'll be able to initiate a registration trial in the first half of 2018. If approval to initiate that trial is granted at that time, we project trial completion as early as 2020. In the meantime, multiple investigator-sponsored trials are ongoing, and we expect to have updates from those trials in the interim. These will support our registration strategy and supply ongoing news flow. Finally, I want to highlight that we continue to have a strong balance sheet. We're disciplined in our spending and will maintain focus on this program to fund its development. As mentioned in the release this morning, with the exception of our CAR-T development, we will focus our near-term resources on PSMA-617. It should be clear as well that our CAR-T development is a highly focused program through a single investigator to develop proof of concept data. It will not require significant company resources to complete that work, dollars or human capital. As for our other pipeline programs, we will explore out-licensing opportunities for those in order to monetize their value. Let me now turn the call over to Dr. Allison Armour, our Chief Medical Officer. By the way, in addition to her experience as a drug developer and medical oncologist, she also has a doctorate in radiobiology. I promise we didn't plan this out when we hired her, but having a chief medical officer with that background will be really valuable as we develop this therapy. Allison will now comment in more detail on the promising clinical data to date and its potential in prostate cancer.
Thanks, Mike. That's great. So, good morning, everyone. I will just talk to some of the points that Mike has already mentioned. As I said, lutetium-177, PSMA-617, is a radioligand therapy targeting PSMA. So PSMA, prostatic surface membrane antigen, is a protein on the surface of the prostate cancer cells. It's a great target because it's expressed about 100 times more commonly on prostate cancer cells than normal tissue, and that will enable us to target the tumour specifically. In addition, we know from our experience in the literature that PSMA tumour expression is often highest in men who have reached the metastatic and castration-resistant phase of their illness. So ECYT has experience in this setting. We have been targeting PSMA-positive prostate cancer with small molecule drug conjugates. So this approach with PSMA-617 is very similar. Instead of attaching a potent chemotherapy warhead, which we normally do, we'll now attach lutetium-177 to the targeting molecule, and that's great because that emits beta as well as gamma irradiation. So lutetium-177 is a good beta emitter. Its half-life is about 6.7 days. That's one of the longest in isotopes that emit beta radiation. And it also emits gamma radiation, which is useful because that can be used for imaging. The path length of 177 lutetium is much longer than alpha emitters, and it gives the ideal characteristics to be able to manage the heterogeneous nature of an established tumour. Since tumour cells up to 1 millimetre from the cell that is taken up, the lutetium PSMA-617 will be irradiated. So this is what we call in radiobiology as the bystander effect. As Mike said, systemic radiotherapy, radioligand therapy, has become established in the field of prostate cancer with the use of radium, but its major disadvantage is that we can only use it in patients who have bone disease. So there is considerable excitement in the prostate field over this new agent. Its ability to treat PSMA-targeted deposits in bone, lymph nodes, soft tissue, and visceral disease would give it a much wider application than radium has at present. And there's been a lot of physician excitement around this product because the potential benefit of treatment, of this kind of treatment to patients in this stage is extremely high. In the field, we feel at the moment that we're in the early phases of realising the tremendous potential of radioligand therapy. With this asset on board, in ECYT, we're at the forefront of this wave of innovation. As Mike mentioned, the physician excitement around this technology is reflected by the fact that so many patients and physicians have been driving the use of this compound in the compassionate use setting and investigator-sponsored trials around the world. We're encouraged with the data that we're seeing from these trials, and it's been consistently favourable. PSMA has actually been in the clinic since 2013. There are currently over 20 peer-reviewed articles in the literature documenting preclinical work, safety, and dosimetry of these agents, as well as efficacy. We know that the doctors have experimented with different doses and schedules, and at the moment, the most established doses are to give 6 or 7 GBq in most of the clinical series. In these clinical series, it's been in the expanded use, the expanded compassionate use program, and all of these patients have received all types of approved therapy that's available to them. Despite this, PSMA-617 has demonstrated a consistent PSA response of greater than 50% between 40 and 60% of the patients in these trials. The RECIST response, when it's measurable in those patients with measurable disease, comes in about 40 to 50%, which is remarkable in the post-chemotherapy setting. It's also been shown to be safe and well tolerated in these patients with minimal toxicity. But moving on, the first prospective clinical trial was just presented by Dr. Michael Hoffman at the European Society of Medical Oncology, or ESMO, in Madrid a few weeks ago. He presented a trial. It was open label and single arm, and a pilot study of 30 castrate-resistant prostate cancer patients, and they received up to 4 cycles between 4 and 8 GBq. The primary endpoints of his study was to examine the safety and efficacy of this compound. He measured the efficacy by the traditional PSA response of greater than 50%. He looked at the bone pain score, quality of life measurements, imaging response in measurable disease, and overall survival. So what did he find? First of all, looking at safety, he found it to be extremely well tolerated. And in fact, the safety and tolerability was consistent with what we'd seen in the previous published retrospective series. The incidence of grades 3 and 4 anemia and neutropenia were only 7%. Grade 3 and 4 thrombocytopenia, only 13%. This hematological toxicity was uncomplicated, meaning that it didn't require therapy. And that means that this compares very favorably to the antibody-based PSMA agent J591, which had been used previously. It showed around a 50% incidence of grade 4 neutropenia and thrombocytopenia, and many patients required hematological support for those adverse events. So in this heavily pretreated population with few therapeutic alternatives, this certainly looks a safe therapy, but also it looks efficacious. Moving on to what he found when he did his efficacy measurements, he found that 57% of patients showed a PSA response of a reduction of at least 50%. 43% of those patients had a PSMA reduction of 80% or more, so a really good biochemical response. When we looked at the overall response rate and imaging defined by RECIST, he found that 71% of patients, their tumours shrank or stayed stable. And the median overall survival in this setting, when all therapy has been exhausted, was 12.7 months. So the safety and efficacy of these data, they also translated into significantly improved quality of life. 37% of patients had improved quality of life, and 43% of patients had a reduction in their pain score. So this trial, which was sponsored by ANSTO, the Australian National Nuclear Research and Development Organisation, was extremely well received by physicians at the conference. And subsequently, they're willing to expand to 50 patients from the original 30, and we expect them to update their results at a medical conference in 2018. Given these encouraging results, ANZUP Clinical Trial Group, that's the Australian and New Zealand Urogenital and Prostate Trial Group, and the University of Sydney are planning a further study about to initiate a 200-patient randomized trial comparing lutetium 117 PSMA 617 to cabazitaxel. So we plan to work closely with our new partners at ABX and the key investigators in prostate cancer globally. That includes very much the innovative investigators at the Peter MacCallum Cancer Centre in Melbourne, as well as the key academic medical centres in Germany and the US. We cannot express how excited we are to contribute to the data from this asset. We ourselves look forward to meeting with the FDA and initiating a trial of our own ultimately, with the aim of bringing PSMA 617 to patients globally. With that, I'll pass the call over to Michael Andrew, who will now outline the lines of our deal, the terms of the deal. Thanks, Mike.
Thank you, Allison. As you've heard from Mike and Allison, we have a unique opportunity to make a meaningful contribution to patients suffering from late-stage prostate cancer, and also to potentially create significant value for Endocyte shareholders. Consequently, we are redeploying capital from existing planned internal programs in order to invest in PSMA 617. Now let me walk you through the terms of the transaction with ABX. This was a competitive licensing process for a registration stage asset with strong data and hundreds of patients. Endocyte was able to become the partner of choice during this process because of our unique capability and experience in this space, which Mike outlined earlier, and a financial term structure that worked well for both parties. Under the terms of the agreement, Endocyte now has exclusive worldwide rights to develop and commercialize PSMA 617. Endocyte paid a $12 million upfront payment to ABX, issued 2 million shares of Endocyte common stock, and issued a warrant to ABX for the purchase of up to 4 million additional shares of Endocyte common stock. Additionally, ABX is eligible for regulatory and commercial milestones of up to $160 million in tiered royalties beginning in the mid-teens. Importantly, the first regulatory milestone, if earned, would not be due until Endocyte submits for regulatory approval. Immediately after closing, approximately 3.3 million of the 4 million warrant shares were exercised by affiliates of ABX, generating total proceeds to Endocyte of approximately $4.6 million. This transaction will cause us to revise our previous cash guidance provided on our last earnings call. On that call, we reported $118 million in cash and equivalents and had guided to a December 31st cash balance of approximately $105 million. Given the upfront payment associated with this transaction, along with some of the incremental investments associated with PSMA 617, our revised end-of-year forecast calls for a cash balance greater than $90 million. I want to reiterate that this deal leaves us with a strong cash position. Regarding our internal programs, as Mike mentioned, we will continue to fund CAR-T through a targeted proof-of-concept study with Dr. Mike Jensen at Seattle Children's Research Institute. We remain excited about that program and its potential utility in solid tumors. Importantly, our investment is relatively modest in order to get to proof of concept in osteosarcoma. I want to reiterate again that we could not be more excited about this transaction with ABX and in helping deliver PSMA 617 to patients. Now I'd like to turn it back over to Sonia to take your questions.
Thank you. Ladies and gentlemen, if you have a question at this time, please press star then 1 on your touch-tone telephone. If your question has been answered or you wish to remove yourself from the queue, please press the pound key. To prevent any background noise, we ask that you please place your line on mute once your question has been stated. And our first question comes from David Nierengarten from Wedbush. Your line is now open.
Hey, David.
That's a new interpretation. Hey, how's it going? So congrats on the deal here. I had a couple questions. First off on just how deeply have you gone into and how do you feel your confidence level is on supply and manufacturing for a radio-labeled peptide here? And then also, when could you get started, or when do you plan to get started on the pivotal study?
Yeah, David, good question. We've actually dug very deeply on, I would argue, every aspect of this transaction and the therapy. The lutetium is, as you might see in publications, is one of the ideal and I think emerging isotopes to be used in this space. And part of that is the unique qualities, nuclear qualities that Allison referred to, but also a big part of it is the manufacturing and availability. It's generally widely available. There are 10 reactors that supply the necessary materials. We have through ABX, who this is their business to manufacture these precursors and in fact participate in labeling. We'll be able to rely on them to a large degree through the clinical trial process. But we're confident that as we've identified the sources of supply, and frankly, as there's another product that's poised for approval in the U.S., just received approval in Europe, Lutathera, AAA compound, is I see that commercial or that market expanding and supply becoming even more widely available. So we're comfortable with that availability. The second part of your question relates to the timing of the trial. As I mentioned in my comments, we've already been in dialogue with the top investigators in this space and statisticians and so on. So we're well on the path to defining that trial, but need to have a discussion with regulators before moving ahead. We believe that we'll be able to initiate that trial in the first half of next year, to be conservative.
And then with regards to the trial that you, I know you haven't spoken yet, but have the are you planning to have a similar design where you confirm and have concordance with the PSA? Sorry if I missed that in the prepared remarks with the PSMA test and imaging.
Yeah, no, it's a really important distinction. One of the things you'll see as you look at the retrospective analysis that was done in most of these patients that were treated outside of Dr. Hoffman's trial, those patients were selected via a PSMA imaging agent that identified PSMA-positive disease. What they didn't do in those studies was to identify the possibility of PSMA-negative disease. And that's, as you know, in our work with selecting PSMA-positive patients, we essentially look for concordance across all disease and to confirm that it's predominantly positive and particularly positive in disease that's progressing. So what Dr. Hoffman did in his study at the Peter MacCallum Center was to do that overlay and exclude patients that had disease that was negative. It turned out it was about 15% of the patients were negative for various reasons, including this concordance issue. In some cases also maybe intensity of the uptake was too low. So it is our intention to use that approach, as we always have, to confirm that not only is there some positive disease, but predominantly positive disease, and we still think that will yield about 80% of the population as appropriate for this kind of therapy.
Got it. Thanks.
Thank you. And again, ladies and gentlemen, if you do have a question at this time, please press star then 1 on your touch-tone telephone. Again, that's star then 1 to ask a question. And I'm showing no questions at this time. I would now like to turn the call back over to Mike Sherman for any closing remarks.
Thanks, Sonia. I know as folks digest this transaction, there will be more questions, and we look forward to communicating with investors and investigators over the coming days and weeks. I want to thank everyone for joining us today. Particularly want to thank the team here at EndoCyte and our partners at ABX. We are incredibly excited to have this asset on board and look forward to meeting with the FDA and initiating a potentially pivotal trial in 2018. Thank you.
Ladies and gentlemen, thank you for participating in today's conference. This concludes today's program. You may all disconnect. Everyone, have a great day.